Good morning, everyone. I'm Steve. I lead the Newcastle University Centre for Cancer, but my research passion is children's brain tumours, and above all, how we can learn about children's brain tumours and try to apply that knowledge to achieve better outcomes for children with brain tumours. So I'm going to show you some research, but I'll try and use that research to get a few key messages over to you, really, about some of the challenges that we deal with, some of the progress that we've made, and where we're really looking to the future. So, we're about to, I'm not sure what the right phrase is, commemorate 100 years of medulloblastoma. So, medulloblastoma was first, it's the most common children's brain tumour, and it was first found in 1925 by this pair, Cushing and Bailey. They made first surgical descriptions of the disease. You can see pencil drawings here that were in the journals 100 years ago. And they made a stab at its incidence, and the incidence is something like 1 per million per year. So, what does 1 million people look like? That's a presidential inauguration in the US. One person in that whole group every year would get a medulloblastoma. So the message is children's cancers are exceptionally rare, thankfully, but do of course create a great clinical problem for us. And then just bringing you through the history, the first treatments for medulloblastoma were to surgically remove the tumor. In addition to that, irradiation is very beneficial in terms of improving outcomes for these patients, and this is going back to the 50s. The first irradiation that was done was simply done by lead shielding, wooden rulers, and marking onto patients the zones that should be irradiated. And that produced, even with those rudimentary techniques, some good survival rates, perhaps 40% of all patients. Then beyond that came the addition of chemotherapy. You will be aware of chemotherapy through the 70s, and again that improved survival rates. The sad news is that fundamentally that hasn't really changed. We know that these tumours are radiosensitive, we know that they're chemosensitive, and through all manner of higher technology approaches, we can now improve anesthetics, we can give high-dose chemotherapy, we can give proton rather than photon therapy, but it's all essentially radiotherapy and chemotherapy. And with that, we're now pushing up to around about 70 to 80% survival for all children with medulloblastoma, which is fantastic. But we're plateauing. We're getting to a point where pushing beyond that 70 to 80% is a real challenge. Don't worry about the graphs too much, but what these show you is that it's not just about treatment. It's also about the side effects of treatment and getting patients out of the other end of their treatment in the best possible condition. What this shows you is that a child that has medulloblastoma and has standard medulloblastoma treatment will typically have a reduced IQ, and that IQ will tend to get worse over the time after they've been treated. Just going beyond IQ, what this really means is that these patients may struggle to go to mainstream schools, they may struggle to leave home, they may struggle to live sort of long, functional lives in the way that you might expect of a child that didn't have medulloblastoma. So that's what it means in a practical sense. So this is our challenge. On one hand, we've got therapies that are efficacious. On the other hand, we've got to deal with this idea of side effects and how we best strike the balance between the two. And this is really where the biology and the research comes in. So what we want to do is to personalize therapy for medulloblastoma. And what we mean by personalizing therapy is being able to place each child into the best diagnostic or risk group and give them the therapy that's best matched to their condition. What personalized medicine isn't is giving every single patient a different therapy. So that's really important. It's really about putting patients into these groups and being able to treat them accordingly. So for a disease like medulloblastoma, it would look something like this. Can we take measurements of the tumor at a very early point in the diagnostic pathway and then use them to individualize therapy? What that really means is that if we can pick out good risk cases, if we can pick out patients that are likely to do very well, we can think about reducing therapy in those cases. If we can pick out the very high risk patients, those that are likely to do badly, we can think about reserving the most intensive treatments for those patients. In the middle, we can start to ask questions of therapies and which therapies might be best. We want also to find all new biology that we could potentially target in the disease. But this essentially is the game that we're trying to play to improve the outlook for patients. We want to place patients into the best groups and then run trials which will allow us, hopefully, to improve survival, but also reduce late effects. Going back to our 1 in a million per year, in Newcastle, for example, probably similarly in Leeds, we see about 5 or 6 cases every year of medulloblastoma. We would never be able to do anything on that alone. So to be able to look at these diseases, we need to work not just locally, but nationally and even internationally to bring together the numbers that we need to do the clinical trials to answer the questions that we care about. And that's exactly what we've been incredibly fortunate to do. So in Newcastle, we've led on biological studies for medulloblastoma in Europe. And this has run right back from the early 90s right up to the present day. And it's really just to show you here that these trials have run. They're essentially the only game in town, which really means that the most contemporary therapies in the disease are being given through these clinical trials. So it's important that as many patients as possible are registered onto clinical trials. And as time's gone on, they've become increasingly complex, involving increasing levels of research. And that's really what I want to tell you about and how that's made a difference. So this is our pathway. This is going all the way back to the year 2000, bringing you right forwards to 2030, and thinking about how we've come along the way. You can see some of the trials we've run. Critically for us, we've had major programmatic funding from Cancer Research UK that have run really for the last 15 years and allowed us to really place the resources into these trials that we've needed to make the discoveries we have and change how we've done things. So just to take you through the clock, going back to the turn of the millennium, what we were doing around that time was using quite rudimentary clinical measures to place these patients into some risk groups. So this is metastatic disease. You're all probably aware of metastases. It's where the tumor spreads from its original location. We would look at them down a microscope, and if they looked a little bit different, we would tend to think that they were a bit more aggressive. And there were some rudimentary biological markers. This is a cancer cell lit up with a particular genetic defect in it. And we would use those to try and start to predict outcomes. This then led us through the tens into a point where we really applied all of the advanced genomics that we now understand. So I'm not going to tell you about this in detail, but just to give you a sense that when I was training, I worked on one gene in about 40 samples, and it took me 3 years. Nowadays, we will look at the whole genome, 25,000 genes, in hundreds of samples. We'll probably get the data back in a couple of weeks. And then we have to spend probably 5 years figuring out what on earth that data set is telling us. So that's research as we understand it today. But by doing this on hundreds of medulloblastoma patients, we're now able to understand that the disease is essentially what we now call an umbrella term. It really represents 4 biological diseases that we can only recognize using biological tests. So I'm going to bring you into the world of boffindom here. One of them is called the wingless. One is called the sonic hedgehog group. It's called the sonic hedgehog group because if you put these genetic changes into fruit flies, their backs go all spiky. And that's why they're called sonic hedgehog. And a group 3 and a group 4. So this is great. We can start to see subdivisions in the disease, but it's only worth doing this, you might argue, if it means something clinically, right? And lo and behold, it does. So this group, the WNT group here, it's about 15% of all medulloblastomas. These are survival curves. And what you can see here is in the WNT group, almost 100% of all patients survive their disease. So this was a major finding for us. And also important that we can pick out patients that do much worse as well. So we can start to think about a disease that was all the same and start to think about putting it into different groups and treating them differently. And this is exactly what we've done over the last 15 years. We're now running clinical trials in different groups. Don't worry about any of the detail here, but essentially we're picking them out using these complex biological tests and treating them according to their disease risk. Really gratifyingly, this is a book that is published by the World Health Organization. So every 7 or 8 years, the WHO, who you've probably heard all about during COVID, they also look after cancers around the world. They publish a book on each group of tumor types that tells you how to diagnose them and what's important clinically. So it's called the WHO classification. In 2016, we moved from having a classification in medulloblastoma that was based on how they look down a microscope to using all of these molecular tests for those subdistinctions. So a really important watershed in the disease, moving to this point where biology is telling us a lot more than what we knew clinically. And the trials look something like this. We now take a tumor, we freeze it at the point that that tumor comes out of surgery, and then we do a whole series of complex tests. We have to arrange that for the country, and in Newcastle we have the National Reference Centre, so every centre in the UK sends their material in real time through diagnosis to us. We take that, we make the readouts that are important and send them back to the centres and to the clinical trials unit. And this all happens within about a 10 to 15 day period after the point of surgery, so that you can make a decision on therapy. And what we're doing essentially is placing them into one trial for the very low risk cases. With them, we're reproducing their radiotherapy. There is a high risk trial where we're trying to figure out the best treatment for the high risk cases. And then in the middle, we have a group where we can start to test different chemotherapies, different radiotherapies to see if they're more or less effective. The other really important thing of taking this biological material is that they go into the Vivo Biobank and we access them for biological studies. And we can then undertake sort of next generation studies based on much better quality material than we had 20 years ago. So we don't just collect brain tumor material, we will collect blood, plasma, cerebral spinal fluid, if there's been a brain tap done. And all of this really fuels future discovery so that we can make it to the next levels. So just to summarize at this point, one disease, 4 groups. This is the Millennium Bridge in Newcastle. I'd like to be able to tell you they lit it up in the honor of medulloblastoma, but they did happen to light it up in the colors that we've given to the medulloblastoma subgroups. But what's really important in research is that things change, and they change constantly. And in many ways, our job as researchers and clinicians is to really decide when we can draw a line in the sand and actually do something in the clinic. So I told you about the sonic hedgehog group. Since the initial calling of the sonic hedgehog group, we can now split sonic hedgehog further into 2 groups, one of which has a survival which is essentially like that other group, the wind group, and one that again does much worse. And a similar deal in what we termed group 3 and 4 disease. We used to think of this as 2 groups. Now we think of it as 8 groups. And this is perhaps the biggest genomic study that's been done of medulloblastoma in the world. We have about 2,000 medulloblastoma cases here. Each dot is a single case. And just to give you a sense of the genomics that we do, essentially this is taking about 400,000 genetic measurements on a given cancer. But what this plot can do is reduce all of those measurements down to a single plot. And then we look at the relatedness between them. So all this is it's an exercise in pattern recognition. If you can pick out patterns and groups that look like they might be separate, then they might be meaningful clinically. And that's exactly what you can see we've done here. They don't all just form one blob where they're all the same in the middle. They do separate out into groups that look different. And when we start to look, we find that they're not just different genomically, they're different clinically, they have different disease features. So taking these groups, they have different ages of presentation, they have different survivals, they have different relationships to other key biological features. So this really brings us to what we can do with group 3 and 4. We now have a group that we can pick out within group 3 and 4 that does incredibly well again, and one that does worse. So we've sort of got to the next level. I showed you WINT that our current trial is based on, where we're treating this group differently. We now have a whole series of patients in group 3 and 4 that do very well, and a whole series within the SONIC group that do very well. When you cut across all of this, we think that we can now reduce therapy intensity for about 40% of all medulloblastomas. And I think coming from a starting point where one size fitted all in terms of therapy, that's a major difference for these children. So this is our latest trial. I won't go into the detail, but we are taking the WINT cases and reducing those even further. We're taking the new favourable risk groups and doing for them what we previously did with the WINT group to take their therapies down. And then we're left with what is now really a high-risk group where we can start to randomise or ask new questions in those. And again, really gratifying, 2022 version of the WHO classifications, all of this new information on the new subgroups have gone into that. But please, please, please remember in this, we are not taking each patient and treating them differently. You know, the natural endpoint of this might be that every patient is a subgroup in its own right. That's not what we want. What we want to do is understand as best as possible, and then, if you like, coalesce the patients back into groups that are meaningful in terms of treatment. And that's exactly what we're doing. And as I say, we're running all of these tests routinely through the Newcastle Centre. So this is just another attempt in Newcastle to replicate one of those biological plots. In this case, it's fireworks over the red time on New Year's Eve. And then I really just wanted to give you a sense of where we're heading. I spoke about lines in the sand. In terms of how we understand biology today, we've worked a lot on DNA and the genes, but we're really now getting to a point where we're working right down at the level of the single cancer cell. And within a cancer, there are not just cancer cells, there are normal cells, and the whole interaction between these can define how cancers behave. So the level of detail we're getting to now in our next trials, is really quite mind-boggling. So one can only anticipate that this will lead to further lines being drawn in the sand in the future as we get more and more findings. So perhaps the most important thing to do is to tell you about the cast of thousands that are needed to do these sorts of work. And you can see some of the stakeholders there, but to me, the stakeholders in our work go right through from the initial patients, parents, and the whole PPI involvement in what we do, through the stakeholders we have to work with to fund the work, right through to the trials organisations that we need to run the trials and do the studies. And then sort of sitting in the middle of that is the actual research team who does the work, who's there on the left. But as I say, we really couldn't do this without people like yourselves and stakeholders right around the world.